The goals of this session are to describe how an action potential in the sarcolemma results finally in force development in the muscle fiber. We'll also ask the question, what are the mechanisms for regulating the force of contraction in skeletal muscle? And we'll consider the different types of muscle fiber that exist. This graph shows the temporal sequence of EC coupling. In all types of muscle, the key event causing contraction is increased intracellular calcium concentration. In the skeletal muscle, the source for that calcium is the sarcoplasmic reticulum on the inside of the cell. The muscle action potential triggers calcium release from the SR in skeletal muscle, and thus the only physiologic stimulus for muscle contraction in the skeletal muscle system is action potentials. So the purpose of this graph is just to show that what comes first is the action potential in the muscle membrane. That is then responsible for an intracellular calcium signal, and it's that calcium acting through troponin C that liberates actin-myosin interaction and allows the development of force. The membrane system of the sarcoplasmic reticulum is enclosed within the cell, and it comprises of the longitudinal tubules, which surround the contractile apparatus and terminate at each end in lateral sacs. The lateral sacs at each end are closely associated with invaginations of the muscle cell plasma membrane called T-tubules. The lateral sacs from two neighboring sarcomeres converge on a T-tubule to form a structure known as a muscle triad. Notice the triads are located at the junction of the A-band and the I-band in every sarcomere. Action potentials in the T-tubules induce calcium release from the lateral sacs. The calcium release mechanism involves voltage sensors in the T-tubule membrane. That voltage sensor is actually an L-type calcium channel, but note calcium is not flowing into the cell from the extracellular fluid here. The calcium channel is simply donating its voltage sensing elements to this mechanism. The voltage sensors respond to depolarization in the T-tubules with a conformational change that results in opening of calcium release channels here in the sarcoplasmic reticulum, known as the ryanodine receptors. Intracellular calcium concentration now increases from about 10 to the minus 8 molar at rest to about 10 to the minus 5 molar during muscle contraction. In order to return a muscle to its relaxed state, calcium reuptake must occur. This reuptake mechanism is located in the longitudinal tubules, where calcium ATPases are located. Following calcium reuptake, most of the stored calcium is found back in the lateral sacs, where it is weakly bound to a protein called calsequestrin. Ready again for the next release signal. The force of skeletal muscle contraction is controlled by altering the firing pattern of motor neurons to the muscle. The effect of increasing action potential frequency to a muscle is known as temporal summation. That's being demonstrated here experimentally, where blue arrows denote an external stimulus to the muscle fiber. At low frequency stimulation, the muscle briefly generates a force, or twitch, and then relaxes, the force trace here being shown at the bottom. However, at high frequency stimulation, the muscle does not have time to relax between stimuli, and contractions fuse into a plateau of active force called tetanic contraction, or tetanus. If greater force of muscle contraction is needed, the number of active motor neurons increases, and this recruitment of additional motor units is called spatial summation. And this is organized according to the so-called size principle, where small motor neurons that reach only a few muscle fibers are more excitable than large motor neurons. The small ones therefore are recruited first. Larger motor neurons then are less excitable, require a stronger stimulus for the central nervous system, and when large motor neurons are recruited, a large number of muscle fibers will be stimulated to produce a very strong contraction. Although they can be further subdivided, there are two major types of muscle fibers: slow twitch or type 1 and fast twitch type 2. This table shows some of the ways in which these two major types of muscle differ. There are expression of different isoforms of myosin and other proteins that accounts for different shortening speeds of these fast and slow twitch fibers. We can see that the slow twitch fibers in general are fatigue resistant. Going along with that property, we see that they engage in oxidative metabolism, They're red in color because they have the oxygen-storing pigment myoglobin. They have abundant mitochondria to support their oxidative metabolism, and their storage of fast sugars and glycogen is low. Whereas our fast-twitch type 2 fibers, these are more powerful. However, they rely on glycolytic metabolism, so they don't have very many mitochondria. They're white in color, partly because they don't have myoglobin, the oxygen-storing pigment, but also because they have a high glycogen content. And whilst they are powerful, they are also very fatigable. Whole muscles are made up of different proportions of slow and fast twitch fibers. For example, postural muscles contain higher proportion of slow twitch fibers because they must maintain tone and resist fatigue. On the other hand, muscles like extraocular muscles are required to make extremely fast, brief movements of the eye, and therefore tend to contain a higher proportion of fast twitch fibers. There are also genetic differences in the general proportions of muscle fiber types that are expressed among individuals, and this accounts in part for a tendency of a person to be either a better sprinter, they have more fast twitch fibers, or to have higher endurance, i.e. they have more slow twitch fibers. Whilst training can have some impact on this, we each have some genetic limitations on which type of athletic performance will be better suited to our bodies. In the next session, we'll introduce muscle mechanics to lay some important groundwork before we get to cardiac mechanics later on.